Thank you all, first of all, for coming out. Thanks for having us come out and talk to you guys. We're pretty excited to share what we're working on, what we're going to work on next. Uh, Plenty of turtle work to be done. There's always projects in the work. Okay, so quick little bio, tell you who I am. I did my undergrad at Towson, knew I liked biology. I wanted to work with animals, the environment. Just didn't really know where that would take me, where I was headed. In my senior year, I got involved with Dr. Kimble and the work that he does with not only box turtles, but the northern map turtle. Got to do a lot of cool field work, go out with kayaks, catch turtles all day. It was a lot of fun. Um, and I always kind of had a soft spot for reptiles, anything scaly, slimy. Um, After graduating, I ended up in an ecotoxicology lab, just a lot of water samples. It was a great experience, learned a lot, but uh, couldn't really scratch that itch with the reptiles. Knew I wanted to get back into science, so I decided to come back to school. Dr. Kimble was the first person I contacted. Of course, he had plenty of things for me to do, one of which is what our talk's going to be, or my portion, um, is working with the eastern box turtles. Uh, these guys have a large contiguous range, the eastern part of the United States, occupying anything from wetlands to fields to forests, sometimes in your backyard. These guys are quite common. Courses, all of our animals do, all of our reptiles, these guys in their natural habitat are serving a lot of roles. It's important to conserve them. A couple to note, these guys do burrow. It's great for the soil as you aerate it, move nutrients around. Some more recent work that's come out, these guys might be really important for dispersing seeds as they mosey around on the forest floor, eating fruits, anything that they can find. As it moves through their digestive tract, they drop it somewhere else. A few studies have found that a seed that passes through a box turtle's gut and out the other end uh, actually germinate better than if they hadn't been eaten by the turtle. So these are just some ecological roles to note, but I think there's a lot more that we don't really know about these guys. Um, and like a lot of our reptiles, they are facing current threats, many of which have to do with us, unfortunately. As we divide their historic contiguous forests up as we build our roads and our neighborhoods and our farmlands. Obviously, those roads lead to roadkill. They're not the fastest creatures. So roadkill is a big problem for them. Poaching, as you'll see in a lot of our pictures, these guys are quite pretty, and they do fetch quite a bit of money in the illegal pet trade. And then disease, of course. There's been a growing concern for uh, disease outbreaks with these guys. They seem to be becoming more common. So that kind of leads us to the microbiome, why we care about it. It's kind of a new topic, especially in wildlife conservation. So, in short, the microbiome is made up of millions of these tiny microbes that live in us, on us, around us. Um, and they turn out to be pretty important and perform a lot of functions in our body. Anything from breaking down our food to producing antibiotics, producing little chemical signals that travel all the way up to your brain and affect your decision making. And one factor that we're kind of interested in is how dynamic this microbiome is, right? It's influenced by a multitude of factors, anything from the host, like your age or your diet. They interact with each other, of course. And what I'm kind of concerned or looking at with this study is environmental factors, right? Temperature, season, the diet that these turtles may have available to them, or any pollutant exposure. So obviously that begs the question, so what's living in the box turtles? What microbes are in these guys? And that's kind of the problem. There's a large gap in knowledge currently about the microbiome of wild eastern box turtles. There's been a few studies of captive individuals. They don't kind of capture the large breadth that I think is needed for a wild microbiome study, capturing a variety of age groups, environments, habitat types, etc. So the goals of my thesis, I think we can kind of break it down into 3 steps. I want to figure out who these microbes are, right? What kind of species. I want to figure out the when and where. Does it matter where a box turtle lives? Does it matter how old it is? And of course, probably the big question is, what are those microbes actually doing in there to improve the health of the turtle potentially? So quickly, we'll go over the study sites where these turtles came from. We have 5 sites in Illinois and 1 in Tennessee. It's probably important to note that all these sites are a mix of habitat quality. And of course, they are not what they used to be, historic old growth forests. You know, these are all affected by human development. They're more like isolated little patches that the turtles, as far as we know, don't move between too much. These turtles are collected via sometimes vets, but mostly dogs. There's a gentleman, his name is John Rucker, trains these dogs, and they're pretty good at going out and finding these box turtles and bring them back to the vets. So over the course of 3 years at these sites, over 500 turtles were collected, and then we take a little Q-tip and swab their cloaca, get all that bacteria on there. Once we have that, we can extract the DNA from there so we can figure out what these microbes are. Okay, so here's what we know so far, and then I think we can get into what we want to learn next. So as we expected, there's a lot of different microbes growing in there, and it's pretty complex. And there seems to be a lot of variation amongst individuals, meaning that if you were to walk outside, find 2 box turtles, pick them both up, their microbiomes probably are pretty different, which is not super unusual, right? That's what we see in humans. There can be 2 people living in the same house, eating the same foods, and there's still a level of variation within the individual. But looking at more of a broad picture on a large scale, we do see some interesting patterns. So now I'm going to talk about what we'll call a core microbiome. And this is kind of a way to simplify things. You know, we're talking about thousands of species of microbes living in there. It gets really complicated really fast. So A common practice in these studies is to establish a core microbiome, which is a subset of these microbes that are just really common and they're pretty abundant. Thinking obviously if lots of turtles have them and they have lots of them, they're probably very important to the host health and their physiology. So here's a breakdown of the now 22 core species. And this is a really good picture of what we can say, we hope to say is the microbiome of a healthy eastern box turtle. There are some pretty interesting things. Obviously, you can see this is turtles captured from Illinois and Tennessee. They share a lot of similarities. We see these big orange circles right there in the middle, with the bulk of those microbes making up the bulk of the microbiome there. But then we see some species that are unique to Tennessee, don't appear in Illinois at all. Notably, that one purple one up there, the fusobacteria, not present in Illinois, which may make sense as these turtles are separated by a large swath of land. Their diet may be different. The level of pollution exposure could be different. But it gets a little bit more interesting as we kind of zoom in geographically here to just Illinois. These are the core microbiomes of turtles captured from each of those Illinois sites. It's probably important to note that some of these sites aren't even 50 miles apart, pretty close. So we weren't really expecting too much variation with the microbiomes between sites, but obviously that's not the case. Most notably, I can point to this Collison site that we're seeing core microbiome only made up of 6 species compared to the others that some reach a dozen. What does that tell us about the Collison site? We're not too sure yet. I'd love the opportunity to get out to those sites myself, but could be a reflection of the habitat, health of the habitat. Another thing that we found is that the microbiome seems to shift quite dramatically over the course of a year. Here we have these circles represent individual turtles and then color coded by the season in which they were captured. And you can definitely see all those blue dots down at the bottom left. Those spring turtles are all grouping together. In this plot, closer 2 circles are, the more related that microbiome is to the other. So further away, more different, closer, similar. So we see some pretty clear trends that turtles' microbiome may shift with seasonal changes, which makes sense, right? Because their diet changes. And also with reptiles, that physiology is quite complex, right? They go through all sorts of changes as they're approaching to overwinter versus their breeding season, warmer weather, colder weather. But this is really important to note because if you're trying to establish kind of a baseline for what a healthy turtle is, you should also be paying attention to what time of year you're catching these turtles. So we've just scratched the surface on this. So really what my main goal next is a question that I have not answered yet is what are those microbes actually doing in those turtles and how are they beneficial? So quick breakdown, what we have been doing so far is just amplicon sequencing of this bacterial DNA, which you don't need to know exactly what that means, but it is a small little fraction of that bacterial DNA. It's really good for telling us what species it is, to different species, how many there are, but it's really just a small little glimpse into that uh, microbial community. So our plans next is to do some metagenomic sequencing, which is sequencing all of that bacterial DNA. And that what that's going to do is paint a much clearer picture of those the functionality, specific genes in those microbes that we can then say these guys are really good for protein synthesis or et cetera, breaking down fibers. Yeah, so that's kind of my next chapter in my master's program. I think switching over here.
Yep.
All right.
Thank you, Jace. Okay. So I'm Steve Kimble. I'm a professor at Towson University, and these pictures here just give a little bit of a representation of various taxa that I've worked on. You can see that there's definitely a herd bias. These days, mostly what I work on are map turtles and the box turtles. Bonus points if you know this one. Very close. Same species complex. It's a crayfish frog in a crayfish burrow here. We don't have them around here, but I worked on them when I was here. So my training has been pretty broad. I started with my bachelor's and master's degree there in Alabama, and in that department you do marine stuff, whether you like it or not. So for my master's, I worked on totally different creature. I worked on sponges, marine sponges that live on mangrove roots. So I did field work in terrible places like the Bahamas and Panama, and I'm being facetious, beautiful places to do some work. And then I went to grad school at Purdue and started working on these guys and have fallen in love with them and have been working on them ever since.
Is that an Ozark Mountain Hellbender or Eastern?
That is an Eastern. That one is from Missouri, though, so close to the Ozarks. But you can tell because it has all of its toes. Has all of its toes. The Ozarks have this weird sort of necrotic tissue thing going on. A lot of them are missing digits or even whole limbs. But this one was posed for me very nicely. Okay, so back to figuring out what we can learn from the microbiome in box turtles. I'm going to tell you about two more projects having to do with box turtles and the microbiome. And the first one has to do with behavior. So most of what we know about this comes from human work, of course, because that's where the money is and the interest is. But the more and more we learn about what's called the gut-brain axis, the kind of scarier it gets. This can almost veer off into a philosophy discussion about what it means to have to have self-will. Because the more we learn that the microbes that live in us and on us, many of them live in us in our guts. It's kind of this weird ecosystem, right? We're outnumbered, first of all, about 10 to 1. Our human cells, an adult has about 1 trillion cells. The microbes are about 10 trillion of them living in us and on us. And they're almost always at sort of the edge. Like they're on our skin, they're in our gut, where the outside, if you stuck your finger all the way down, that would still be sort of the outside, interacts with your insides. And they're doing lots of important things in there, like Jace explained earlier, like helping us make vitamins and things like this. So we don't really survive very well if we don't have these guys living in us. But they're also releasing all these other chemicals and things that can go into our bloodstream and affect our brains. So the more we learn about this, and again, most of this work has been in humans, the kind of scarier it gets. You are definitely what you eat. So this can affect our behavior. And scientists started to turn away just from humans, and everywhere we look, pretty much every large organism has a microbiome, and everywhere we look, it seems to be affected in some way by their behavior, seems to be affected in some way by their microbiome, or the other way around. It's not at all clear sometimes what those differences are. Here's an example. Again, in humans, because we know the most about this. This is from an experiment. So it turns out that people who are on the autism spectrum, they tend to have different microbiomes than people who are not on the spectrum. So one obvious treatment has been to move microbes from the people who don't have the autistic symptoms to people who do. And when you do those sorts of experiments, you can see that when it's sort of simplified into these three categories of severe symptoms, mild to moderate, and minimal to no symptoms, here's week 0. This is when the treatment has taken place. So these are microbial transplants. They're literally taking microbes from a person who does not have autistic symptoms and moving them into a person who does. You can see after 8 weeks, and even long after the treatments are over, up to 2 years later, there are definitely some measurable effects on autism, which is a syndrome of many different symptoms, including a lot of behaviors. Okay, like I said, there's been a little bit of work outside of humans. Here is a cool cartoon drawn about a zebrafish experiment. Zebrafish are often used in genetics because they are easy to grow. Has anybody raised any zebrafish? They're super easy to grow. They're really hard to kill. And you can grow huge numbers of them. They reproduce really quickly. So they're easy to study in a lab. Box turtles are terrible for this because it takes forever for them to mature. So in this experiment, they were using zebrafish. And what they did is they managed to grow up some of the zebrafish without a microbiome. They basically gave them enough antibiotics that they didn't have any microbes living in them from the egg stage. And then they grew some up that were normal. They still had their regular microbiome. And then they put them in these fish tanks next to each other, and their behaviors were much different. And these orange tracks is tracing where these zebrafish are going. These zebrafish were grown with no microbiome for the first week of their life, and they are totally uninterested in each other. So we're looking down on 2 tanks, and these fish are not at all interested in each other. They're not spending any time along the glass trying to get to that other fish. These zebrafish were grown up normally, and they spend most of their time trying to hang out with the other zebrafish that they can see running the other side of that glass. So this seems to suggest that there's something about the microbiome that is affecting the way they are developing. So again, we probably couldn't survive very well if we were all sterile, if we didn't have these microbes, not just us, but any species that we've looked at in depth so far. So of course, this is a talk about turtles. And I think this is going to take me to another site, but we can watch this cool little video that you may have seen. This is just a little bit of BBC footage showing a box turtle and how it responds. Hang on, it's not showing you the actual video. Technical difficulties. It's just like I'm teaching. There's always something. So if I can get this slideshow to minimize, then I think maybe Zoomers can see the video. Oh, but of course it opened it in a different browser.
The people in Zoom are seeing what's on the projector screen.
Yep.
Sharing screen 2. So I think you might have to share screen 1. I don't know.
Or if I can
drag it over to screen
2. Yeah. There we go.
Use it for professional.
Okay. All right, that is a behavior, right? That is a movement that the turtles are doing to avoid being eaten by that hungry raccoon. Okay, so I mentioned that because so far it's been really hard to describe behaviors in turtles, but we've done a little bit of work showing that turtles actually can have what you can call a personality. If your definition of personality is some sort of repeated behavior, like hiding from a raccoon, it's repeatable. So you pick up the same turtle twice and it acts in the same way. So you saw that turtle, in my experience, would be a relatively bold turtle. Has anybody ever seen a live box turtle in the woods? And by the time you see it or get to it, it is totally closed up. That's a more shy turtle. So, it can be clear what a really bold turtle is and a really shy turtle is. We wanted to quantify this. So in this particular study, what we did was we went out and we found turtles. We put them in a black bag, no lie, to sort of reset their experience. And then we took them out of the bag after 3 minutes, put them on the ground, walked away and hid ourselves and started a stopwatch. And some turtles are bold in that they'll come out pretty much right away. Their head comes back out and they want to see what's happening, as if they've forgotten that they are box turtles and there might be dangers out there that their shell could protect them from. Others are much shyer, and even we stopped the stopwatch after 10 minutes. Some of them would still not sticking their heads out. They're just going to wait it out. And it turns out if you pick up the same turtles over and over, they always do the same thing. They can be a little affected by temperature, but not too much. So each of these rows is a different site from Massachusetts, Michigan, Indiana. There's a Maryland site here where we collected some data. So box turtles definitely have this personality. So we were interested to see if this personality trait has any relationship with the microbiome. So if you think about it from the point of natural selection on box turtles, there can be arguments for why you might want to be a bold turtle and why you might want to be a shy turtle. Based on that video, why might it be good to be a shy turtle? Right. A good way to escape from predators is just hide in there and be shy. It's a turtle. It doesn't need to eat for the next hour or whatever, right? What are some good reasons why you might do better if you're a bold turtle? Yeah, when you're all boxed up, you might be missing other opportunities like food. Maybe an earthworm went by while you were boxed up and you didn't
get across the highway.
There may be something to that too, about because some box turtles, when a car drives by, they box up, and of course then they're just sitting there waiting for the next car to come by, whereas a bold turtle might fare differently in that kind of scenario. So all right, so personality traits like this in many taxa have been found to have some relationship with the microbiome. We have to be careful here in not saying which one causes which. Personality may cause the behavior, or the behavior may cause the microbiome. But it was worth exploring. So this first project I'll tell you about had several components. The first thing we wanted to do was see how to explore a little bit more about this behavior. And I'm sorry, the print is kind of small, but what this shows you is a bunch of different traits that you could measure. These are in humans, and how heritable they are. So this first one is eye color. That is almost 100% heritable. So that depends almost entirely on the color of the eyes of your parents. Whereas something like, and I don't know how they're quantifying things like depression and trust, those have lower heritability. And you can do these kinds of studies with twins and with families and things like this. You just track how heritable those things are. And then So we wanted so the first thing we have to do is come up with those kinds of numbers for this kind of personality in box turtles. We did this particular project at Ali Memorial Woods, which is a long-term box turtle site. The box turtles here at the spot in Indiana have been studied since the 1950s, which isn't very long for a box turtle population. And this paper is from 1987, and they are showing us how many turtles they are capturing here. And then using some math, they're trying to figure out how many are actually out there. So you go out multiple times, and if you are always capturing the same turtles, then you know that you pretty much have captured all the turtles there. If you're always capturing new turtles, then you know your population is probably much larger than the ones that you've actually seen. So that's where these numbers come from. And just like at almost every long-term box turtle site, the numbers have been in decline since the 70s or so. That's the same here, a couple of long-term sites here in Maryland, this site, et cetera, et cetera. This work was done in collaboration with Brad Carlson. He's a faculty member at Wabash College. It's this tiny little men's-only college in Indiana, and they own this site. So the methods are, I don't really need to show them to you again because you've already heard about them. You go out, you find the turtles with dogs, you pick them up, and then you do cloacal swabbing to get the bacteria on your Q-tip, and you sequence that. And then you get a unique DNA fingerprint, just like you've seen on daytime TV, trying to figure out who's related to whom or who was the daddy. It's the same trick for any organism that has DNA, which is anything alive, pretty much. So we went out and picked up a bunch of different turtles for this study. And I think it's close to 100 different turtles for this study. And without knowing who's related to whom, you can put all that DNA sequence information into the computer, and the computer will figure out who's related to whom, if you're lucky. So this slide is also here to remind you from your genetics class that you might have had a long time ago. You remember Mendel and his pea plants? So he figured out long before there was DNA sequencing that organisms like pea plants and turtles and us, remember we have 2 copies of most of our genes, one we get from mom and one we get from dad. So this is a hypothetical family tree of turtles here. Here's a bold turtle and a shy turtle. And here are their genotypes. So big R, little r. You might remember the wrinkled peas and round peas and tall plants and short plants. This is a hypothetical tree because we do not know exactly for this particular trait. We don't know what the genes are, and we don't know what the alleles are. But we do know that just like humans and just like pea plants, box turtles have 2 copies of most genes, one from mom, one from dad. And so if you have a big R parent and a little r parent, you get a bunch of, anybody remember that word? They have 2 different versions. It's called a heterozygote. Yep. That'll come up again in a minute on the quiz. I'm joking, no quiz. Okay, so this may be from your textbook. These are Mendel's peas here. He was talking about wrinkled and round. Same thing. So we did a couple of we were hoping to do a couple of different things with the DNA information from all those turtles. One, figure out who's related to whom. And because we know which turtles are bold and which are shy, we can see if boldness or shyness runs in families. And then the other thing we wanted to do is see if there was anything to the heterozygosity across these turtle genomes. What we find sometimes is something like this. This is a study from somebody else at Towson comparing the heterozygosity. Remember, I just mentioned what the heterozygosity is across the entire golden eagle genome. And what they found was that in adults, the heterozygosity was higher than it is in chicks. Sometimes we assume heterozygosity is better because it gives you more genetic diversity. So this is really important for things like immune loci, because when a new disease shows up, if you're heterozygous, you might have more sort of immune power to fight that off. And we see this pretty strongly in this case. So these are all the chicks in the nest, and they have some amount of heterozygosity. But the adults, these are the ones that have survived chickhood to being adults, have higher heterozygosity. That suggests that chicks that are not as heterozygous probably died and didn't make it into adulthood. So at least in this system, it's great to be heterozygous. Maybe there's something like that here in these turtles. All right, so this is where we hit our first snafu, but we learned a bunch of stuff about box turtles by doing this particular thing. Here is just a distribution of all the relatedness values we found. So we had something like 92 turtles, and what you do is you get the computer, you don't do this by hand, you get the computer to compare every turtle to every other turtle and see how closely they're related. And most of them are related by about 0, which means that they're unrelated. If you have a relatedness of about 0.5, that's like parents and children, or full siblings have a relatedness of about 0.5. Among these 92 turtles we sampled at this site, almost nobody was related to anybody else. So we couldn't really do this trick to see if this is a heritable thing. With the heterozygosity, so instead of chicks and adults, now what you're seeing is bold and shy turtles. We found no statistically significant difference between those two in heterozygosity. So, and then the third thing we did, because this is a microbiome talk, is we also swabbed those turtles, and we found a bunch of weird stuff. So we took oral swabs in addition to the cloacal swabs, and we got long lists of things that live in there. And then we also got long lists of things that these things that live in the turtles are predicted to do. So I put this picture up here to remind me that they're just all these lists of things that live in there. And then the computer can also tell us which ones are present just in the bold turtles and which ones are present just in the shy turtles. And we got a bunch of stuff that we haven't interpreted yet. For example, this creature is present in all the bold turtles and not the shy turtles, and we don't know much about what it's doing in there. This creature has been found in things like extremophiles. Those are the bacteria that can live in hot springs. They can survive radiation. You can take them to space and they will survive. We have no idea what that has to do with being bold or shy in box turtles. But so, okay, we'll put that on the list of things to do. The next grad student will get that. We'll get those kinds of projects. It really is a weird list. It'll be like, this particular bacterium is not very well known, but it was first described in marine sediments from Antarctica. And yet we found it inside of a box turtle in Indiana. And this is what you get for studying a non-model species. If we were doing this in humans, we'd know a lot more about what those creatures are doing. But in turtles, we don't know so much. And then I mentioned the computer will also tell you, using these big databases, not only who's in there, but what they are doing. So it again gives us these long lists of some things that are just really hard to figure out. Like, does anybody recognize this word atrazine? It's glyphosate. It's a pesticide. This is in a area that's surrounded by agriculture, so it's not that surprising that this is showing up in these box turtles. But why this would show up as a difference between bold and shy turtles, we don't quite know. It could be that maybe the bold turtles, because they're bold, are going out into those cornfields and being exposed to atrazine, and the shy turtles are not crossing the road. They're staying in the nature preserve. The nature preserve is just kind of on the side of a stream, and it is a nature preserve where box turtles live because it's too steep to have ever been cleared for corn or soybeans. So they're surrounded by agriculture. Primary bile acid biosynthesis. This has been studied in mice in terms of behavior, and mice that have lower biosynthesis tend to be more chill mice. It's thought because they have vitamin deficiencies. So maybe there's something to do with the diet in the box turtles. Okay, again, we'll put this on the list of another student to track down. There's definitely a lot to be learned. So our take homes from that are that basically we have a lot to learn about these turtles. We're still at the point where it's hard to make like conservation recommendations based on what we've learned. However, the fact that despite the fact that this is a small and probably shrinking population, it appears that all those turtles are unrelated to each other. This wasn't the primary goal of the research, but that's a crazy story.
How do you get a population of turtles that aren't related to each other? I mean,
Good, great
male
question.
and female, they have kids. How are the kids not related to the parents?
It could be that the kids are not surviving, so we're not measuring those. It could be that the males or the females are dispersing and going somewhere else, maybe going into the cornfields and then getting plowed up or something. It could be that they're all just old timers. You know, box turtles, we know they can live at least 100 years in the wild. Probably not the average, but they can do it. So maybe all the turtles that we pick up are just all really old from when that habitat was more intact. Or, you know, we do see them on roads. So there probably is some natural level of box turtles moving, perhaps to the next nature preserve. You know, this is along a creek and there is some wooded habitat along the creek. So they could be doing that, or they could be getting washed down during flood events. They're, you know, they're in the family of swimming turtles. They themselves are not particularly aquatic, but they can swim. So maybe they're getting washed down. It's in a river bottom area. Yes, we too are still shaking our heads and scratching our heads over this kind of thing because it's really hard to explain with what we know about the natural history and about the history of this site. So that's a cool thing to mull on, right? We'll add that to the list of future projects to do. And then the fact that we find basically no genetic difference as far as we can tell between the bold and the shy turtles, at least across the whole genome. That's how this is measured. Might suggest that there are just one or two genes that are responsible for being a bold or shy turtle. And so we're just not looking at the right gene yet. Stephen, did you have a question?
Yeah. So I had two kind of questions. So for one, like if there are 92 unrelated turtles, how big do you think that the population in that area is? And also, like, how big in land mass is the?
I think the preserve is 200 acres. But there is some wooded area outside of the reserve. And of course, the turtles don't respect political boundaries like reserve boundaries. So if they're able to disperse farther, then it's hard to say how big the population is in terms of geography. We're working on that number in terms of estimating how many are in the population, because again, Brad Carlson, whose picture you saw earlier, he's trying to get an update. These numbers are from 1983. No one has tried to recalculate this since 1983. And I've seen the email, and I think it's about the same, but you get estimates, and it'll say, oh, there are approximately 74 turtles here, plus or minus 50. Kind of like when you hear about political polling, they have errors, right? So it's not like two. So they're clearly turtles still there that Brad's picking up, so they haven't been wiped out. But we don't see a lot of younger age classes, suggesting that they're maybe not reproducing very well. So this number probably is not increased. But what we see at other long-term box turtle sites is a reduction since the 1950s or 40s of about 70%, and then they've stabilized. We've seen that at Patuxent Refuge down near DC, see that at Jug Bay down farther down on Patuxent, Prince George's County.
When you find some box turtles, they're actually worn down smooth, you know, they're very, very old.
Yep.
You find a juvenile turtle, it's not adult size yet, you know, it's young. For all those turtles in between, is there any way to predict the age?
Not so far. That would be, so many people would like to have the answer to that. marking and recapturing them is one of the few good ways. And that's why another reason these long-term study sites are so important, because we can still pick up box turtles occasionally that were marked in the 70s or 80s, and we can still read their tag and know how long they've been there at a minimum. They might have been caught as adults too, but we might know that they're at least 70 years old. Now that we have these genetic tools, of course, you could pick up a turtle and sort of DNA fingerprint it. But again, you would have had a capture some time ago, or it would be really nice if they were related to each other, because then you could put together these family trees, and you could learn a lot about inbreeding and who's related to whom. We would love to learn if some turtles are really good at having children and grandchildren, and some are not. But when they keep confounding us by being totally unrelated, it makes that kind of stuff harder.
So if these turtles they breed and they're born and they grow for a while, and then they disperse, like nobody knows when or where or how.
We know very little about that young stage because they're so hard to put like radio transmitters on. They're so small. The technology is helping with that. What few studies we know about, they tend to go, at least in their first year or two, they don't go but about 100 meters or so. But then between like maybe age 2 or 3, they start to get hard. Not that they couldn't be eaten by a raccoon, but they can at least come up above the leaves and then maybe disperse. We know almost nothing about that life stage. And then once
that's the age at which they can close up, right?
They can start to close up, and they're not squishy like they are when they're hatchlings. We just don't know much about that life stage. It's the same with sea turtles. These sort of mystery years between hatchlings and adulthood, we just don't know a bunch about what they're doing. But the technology is getting there. Transmitters are getting smaller and smaller and lighter, which is starting to help with that. But then sometimes we have these adults that also seem to be on a mission, and they're on the move too for unknown reasons.
Speaking of like technology and stuff, do you know how they like looked at the genome of or determined how related different individuals were in this collection compared to like how they do it now? Because I imagine it's got to be like far more accurate now. They're using like mRNA.
Yeah, at that time they had no way, without a pedigree, without a family tree, they had no way of knowing who's related to whom.
Then you said like 92 were unrelated. What they thought?
Those are from current samples, so from
okay.
3 years ago or so.
Oh wow. Okay. Sorry, I thought it was from the old.
Yeah. In some places, you can go back to the old ones if they've been like preserved in a natural history collection. You can go and get DNA out of them, and you could do that study. You just need to have a collection, somebody who thought 40, 50 years ago to collect a bunch of turtles. Stay tuned, because as you know, in the basement of Towson University, there are several hundred box turtle shells from around here, and you can get DNA out of those, and a future student is going to ask some of those questions. How have the genetics changed from now to back then?
Well, most, well, I've read too, most females, when they mate with several males. Most of the eggs have different fathers anyway.
Yep. So they might be.
Lead into
Yep.
what's going on too.
Half
and plus, they can store sperm these 5 years. So.
Yep.
You never know. You don't know.
It's a crazy system. In theory, the DNA can resolve all those questions. It can tell us how many fathers are involved in one particular clutch. It can tell us about sperm storage and things like that. But it appears that you need a really large sample size. Apparently 92 is not enough, which is a really big sample size for box turtles. That's a lot of work. Okay, last project that we'll talk about with box turtles and microbiome has to do with keeping with the differences between turtles that are in like a zoo situation and those that are not. So this comparison has been done a lot, mostly in primates. And what you tend to see when an organism goes from being free ranging to being in, say, a zoo or an aquarium, is that the microbiome changes. Remember early on, Jason saw that, showed you that slide showing you all the different ways that a microbiome can change. And one of those, and several of those are relevant here, is what's different in captivity than in a free ranging population. Say box turtles. Diet's different. Temperatures may be different.
Lighting, UV
Lighting. Right. All those are different. And if you're right, yep, the dirt may be different. So everything in the environment, lots of things in the environment can be different when you take an organism from the wild and bring it into, say, a zoo or aquarium situation. So here is just a typical study in, I think this was in non-human primates, showing that a couple of things, the captive and the wild ones are very different in terms of the richness and the community of all those things living in there, and statistically significant. But this is for primates, and that's where, of course, it's been studied the most, in particular primates that are in zoos, and they come into a lot of contact, maybe not direct contact with the primates themselves, but they share a lot of things in their environment. The same dirt, same lighting, same proximity, so they can be sharing a lot of the same microbes.
So how does that relate to their general overall health and longevity?
Great question. That's what we're trying to get at with this particular.
Because some things seem to live longer in the wild than in captivity. Right. And other things, in captivity, they live twice as long than in the wild.
Right.
So and it seems contradictory.
And what's really difficult to pick out is what is it that makes the difference? Is it the diet? Is it the amount of space they have? Is it something else that affects their well-being? We suggest that the microbiome has some role in that. That's why we did this particular study. So the Maryland Zoo, like many zoos, especially accredited zoos, are really interested in treating their collection animals in the most natural way as possible. Now, you can't give an elephant as much space as they might have out in the wild, but with something like a box turtle, you can probably approximate that pretty well. So this is a picture that Kat took of the box turtle enclosure at the Maryland Zoo. And this is to give you a feel for how naturalistic it is. So the turtles are back here. Apparently this used to be a ground prairie dog habitat. So the turtles are confined here, and there are these little bubbles where my kids like to stick their heads up and look around for turtles. And there are these walls here which keep the turtles in. There's a little pond right down here and a glass wall. So these box turtles can't really get out. But there's also a collection of free-ranging box turtles, same species, same subspecies, that are just outside here. So the same forest, the same weather, probably same dirt. Snakes will go between them. Frogs probably go between them. Certainly lots of invertebrates go between them. So the environments are pretty similar. They do have a different diet in here because the diet in here is supplemented for these turtles. They have veterinary care, which to some degrees the turtles out there on the other side of the wall may or may not. And the density is different. They're 22 turtles or something in this enclosure, or at least when we sampled these, which is a much higher density than you would see in the wild. So this is a kind of a nice natural experiment because they're pretty similar, by which I mean inside the enclosure and outside. So this is a great place to ask that question, like what is different or similar about the microbiome for these turtles. And I should also mention that the turtles that are outside of the collection are often encountered by zoo personnel and even by visitors to the zoo. And so those guys were captured as they were encountered and swabbed, just like in that picture that Jace showed you earlier. Same methods, swab the turtles, extract the DNA, and then identify who's living in there. And a similar kind of figure, just showing you the proportions of the different phyla of bacteria that we found both in the zoo and in the free-ranging population. And this tells you what the phyla are, but what's important here is that the colors and the proportions are pretty similar. Okay, so these are great for visuals, and you can kind of eyeball them, but we like to have p-values and be able to say things like statistically significant and things like that. So we did some of that too. And earlier I teased you with this idea that often in captivity, organisms will have a lower richness. Richness is just, well, one way to measure richness is just to count the number of species of bacteria. The captive ones in this scenario seem to have fewer species of bacteria in captivity than they would in the wild. Generally, that's considered to be a bad thing. The more diverse an ecosystem is, whether it's the guys who live in your gut or the herps that live out in the Maryland landscape, the more species they are, the more stable that ecosystem is. So anyway, we did some statistics and we found that the contrary to what we had predicted, the diversity here in the zoo is actually significantly higher than it is in the turtles that live right outside of the collection. So again, same woods, same weather, probably the same dirt, lots of same creatures and trees and such in both of the environments, but the richness seems to be higher inside of those turtles that are in the collection. So I think the main take home we get from this particular study is that the habitat here does a pretty good job of imitating what the turtles would be encountering in the wild. Perhaps even better, as judging by that richness being a little higher in captivity. So good job, Maryland Zoo, for the box turtles at least. They're doing a good job providing an care that seems to mirror, if not improve, on what's happening out there. Okay, so those are 3 projects that are in the works, or in one of them has been published, and one of them is still in the works, and one of them is somewhere in between, where we're trying to learn what we can about the microbiomes in box turtles, and all of this with some sort of conservation bent. In the end, what we like to do is be able to say to managers at zoos or aquaria or DNRs or whatever, this is something you can do to help the species you're charged with protecting. And this against a backdrop of turtles, which are poorly understood in most aspects of biology, but in particular for microbiome work. We're really hopeful because the more we learn about microbiome work in humans, the more important it seems to be. So if we ever get to personalized medicine, it is definitely going to consider the microbiome component. Your doctor may ask you for a stool sample every year when you go in for your physical, and they can sequence it and tell you all kinds of things about what might be inflamed and what might your proclivity be for certain kinds of cancers or irritable bowel syndrome or whatever. So we're hopeful that this can provide some conservation pointers for species managers. So far, we see the box turtles have a highly varied microbiome among individuals and between sites, as you saw from Jace's maps. Some similarities, but a lot of differences between Tennessee and Illinois turtles. But even within Illinois, and some of those parks, if you see on the map, are very close to each other, 50 miles or so, and yet they have different microbiomes.
Have you recaptured turtles and found any significant difference from year to year or years to years?
You want to address that one?
Great question. We have a couple recaptures from 2013 to 2015, so small window of time, but there's a bunch of variation amongst individuals, but an individual's microbiome is stable through time. So if you catch it in 2013, catch it in 2015, it'll be pretty similar. I would love to get more current samples because it's only 2 years. Get a recapture, we might. The same team will go out this spring and summer, same sites, and we're just kind of crossing our fingers that we get it recaptured. So we can do that in a decade now. See how stable that is over 10 years or 11 years. Yeah.
So ask that question again in about a year. we'll sequenced all the. Hopefully, we'll get some recaptures from it. But 10 years for a box turtle is not at all unusual if we do get some recaptures. Some of these sites are just little like county parks, a few hundred acres surrounded by corn and soybeans, so they should be kind of pinned in there.
Mushroom hunting, I found the same box turtle 4 times in 5 years. I haven't seen it for the last probably 8, but that was like crazy to me.
Yep. Some of them have really small home ranges too, like the size of this room. They seem to get everything they need from a really small home range. So box turtles have a highly varied microbiome. We've seen that across space at least. And Jay showed you a picture where it seems that their spring microbiome is different than the summer fall one. However, we know that if you test the same turtle over several years, they seem to have a pretty conserved microbiome too, which is again what we see in humans, and again might be really important for figuring out what is a healthy microbiome for a particular turtle or particular population. Because eventually, it would be nice if we could say, here's what a normal turtle microbiome profile looks like, and here's one that looks like it's sick or something like that. Functions are still poorly understood or poorly predicted. So you saw that like bile acid synthetase and atrazine metabolism. We don't really know much about what's happening there. Or I put poorly predicted because right now the tools that we have are just a bunch of computer software that's kind of based on expert opinion. So you sequence these guys and you feed it into the computer, and there are databases that say, well, if you have species X, it might be associated with atrazine degradation. Those predictions might not be so good. So what Jace is proposing doing, where we sequence the entire bacterial genome instead of just one locus, should help with this stuff a lot. For example, think about E. coli, right? The same species of bacteria lives in our guts, helps us metabolize vitamins. We need it, or we don't have this particular vitamin. But it's also the same species that if you get the wrong batch of hamburger, will give you, might even kill you. It's the same species, but the difference is in the genome somewhere, in different genes. And right now, where the state of the science is right now, we're not sequencing that part. But what Jace is proposing to do is he would be able to sequence those genes that are making one version beneficial and one version pathogenic. So we should learn a lot more about their function using that. This is all new. We're figuring out how we get all that information because you get gigabytes of data, A's and T's and C's and G's. So we got to get the biology out of that. And echoed, the number 3 echoes that. We see a lot of things in there that we just don't know what they're doing in there. All we know about them is they're extremophiles. You put the name into Wikipedia and it'll say, first discovered in the Mars lunar lander before it was launched. No lie, that's been done. And it just means that it's probably pretty common. So it's in the butt of a turtle, but also on a lunar lander in a clean room. But we just don't know much about it because it doesn't end up in human samples. And that's where most of the work has been done. So after we learn a lot more about them, then we can start doing more of this. So far we've been able to say to the Maryland Zoo, good job, keep doing what you're doing, basically, with this. But the DNR would like to know, well, what can we learn about the microbiome for helping preserve these populations of box turtles? Here in Maryland, we have a couple populations that have been monitored for decades, and they're in decline, even though the habitat is protected. There hasn't been any known disease outbreak, no known poaching. There's no development, so no houses, roads, things like that. And yet the populations are crashing for reasons we don't understand. Might be some pollutant, might be forever chemicals or something like that, but the microbiome might be able to give us some clues to what's happening. So that leads us pretty directly to what we're going to do next. You've already heard some about that. What are they doing in there? Jace's project will help figure out. We'll learn a lot more about the genomes of those creatures. This would be nice to know, what's healthy and what's unhealthy. We see a lot of variation among individuals, just as in humans, but can we figure out what are healthy microbes to have in there and what are not healthy microbes? How are they affected by turtle environments? So we're trying to get information on the pollutants they're exposed to and the weather and the habitat type and all kinds of stuff. I mean, all kinds of things in the environment, right? Not just the different diets and the different weather. Some of these populations, of course, have had disease outbreaks like ranavirus outbreaks. Does that have anything to, can we see the effects of that in the microbiome? Would it help predict the next outbreak? And then forensic applications would be really useful. So, you might have heard about confiscations. Right now, this species is the number one poached turtle in North America right now. It's usually heading for markets in Southeast Asia, but it'll get found at an airport in New York or even in Hong Kong, and the genetics are not very useful about telling us where they came from. It would be great if maybe the microbiome could tell us where they came from. Because Jace has already seen that they're different. The microbiome is different in each little state park. Maybe that could tell us where these turtles came from, and maybe we can take them back. And then that would also help prosecute. That potentially could be evidence in court for saying, yes, this turtle came from this site, and therefore this person who has been accused of this might build a case against them. Because when you're a poacher, you can just say, oh, well, these are captive bred. They were not wild caught. But maybe there can be some forensic applications to help get to the bottom of that. No, that's probably not going to be. Okay, maybe I can show you this video. This is a video of the turtle dogs at work. It's just a fun video to watch. And it also gives you a little feel for what the habitat is like at that Ali Memorial Woods site. Now, the turtle dogs are great. They can find like 10 times more turtles per hour than humans. But in this particular case, I saw the turtle first. So those are Boykin Spaniels. They're bird dogs that John Rucker has. I guess we can't hear the audio, but I'm standing over there saying, I see the turtle, and the turtle was right at my feet. But you can see how the dogs work. And you can see what the habitat is like at Ali Memorial Woods. It's all this old, mature stuff, not very good for turtle breeding. There's not a lot of sunny, sandy stuff. And they're so excited when they find it. And because they're bird dogs, they have a really soft mouth. They pick up the turtles and bring them to us. But because they're trained as bird dogs, they don't really hurt the turtles. And their mouths have been swabbed, dogs' mouths have been swabbed for disease. We know that they're not transmitting this. In case anybody's worried about. Anything else? I think that's it. We'll take any questions you have. Thank you.
Thank you so much.
I have one question.
What's that?
Do you feel like our science is adequate to head off this decline? What's your gut feeling on that?
My gut feeling about if the science can rescue us from the decline? I'm cautiously optimistic. I think there's a lot to be learned in the turtle DNA and a lot to be learned in the DNA of all these microbes that live on it. One thing learning about microbiomes teaches you is you have to stop thinking about humans or turtles or any of the other large creatures in this room as a single organism. They're actually whole communities of stuff doing what whole communities do, which is respond to changes in their environment. So I'm cautiously optimistic that this kind of science can help. But I also am thinking about working on a project that would help with like reducing roadkill crossing too, because that's also a big threat for turtles too. So I think if we keep doing what you're doing here at the museum, training the next generation, the next one thing that I get privileged to do is to teach college students and grad students, and they have much different views on the world, in particular about conservation, than our generation has. So I think they'll be much better and pay attention more to things like right now there's a study going on trying to identify wildlife crossing hotspots on roads in Maryland. So and eventually it will result in our website. You can go and see where the hotspots are it's going to be mostly deer because those are the ones that are so expensive for insurance companies, but other creatures too. A lot of tools are coming online to help with that.
Yeah, I got my own opinion about the reduction in population. It's a lot of raccoon predation, a lot of predation on nests and everything. Why we're not seeing the recruitment of young? Because I fish a lot and fish new ponds and stuff like that. I went to one pond this year. Every nest was raided. I mean, I found like 14 turtle nests. As soon as they're laid, they're gone. They're gone.
First night. Yep.
And raccoons thrive off humans. And plus people don't fur trap anymore, really don't hunt raccoons in the area, so the population's crazy. Like this fall, going from Perry Hall to Bel Air, there's like 10 raccoons dead on the road like every morning. So it tells you the population's super high, because turtles ain't got a chance with raccoons.
Yep. The other species I mostly study, we mostly study in our lab, is the map turtles. And it's kind of the reverse in this weird way up at Port Deposit on the Susquehanna River. It's this urban site with lots of dumpsters, lots of people, lots of restaurants. And yet, and so there's a nesting site right in town, no raccoon predation. And there's a nesting site outside of town about 5 kilometers. And it's like that. It's every morning, every night that's a snack.
I'm bass fisher, fish 25 years. I'm there all the time. There's a lot of mink.
Oh yeah,
a lot of mink too. And Conowingo Pool is loaded with map turtles. Like Broad Creek, loaded with them. You see 20 of them sitting on the wall, loaded with northern maps. So I don't know if you study the upper pool of Broad Creek, but there's a ton of them in there. I rarely see anything below the dam, like Port Deposit.
No, they're down there.
Very few, but most of them I see most of them in Broad Creek.
Yeah, we see a lot in Broad Creek, a lot of cross in Conowingo Creek. The nesting up there seems to be more dispersed, not one big, like below the dam, there's one big nesting area, maybe 2, maybe 3. So they're concentrated, and the raccoons learn from their parents how to dig them up. And so that spells trouble. Up above the dam, the mink.
And up top is like, there's not much nesting areas up top either, because it's mostly sheer rock.
It must happen, because they're probably not crossing the dam.
I see a lot of babies up there too.
Okay.
Adults. The females are way larger than males.
Little guys.
But that's like we're at like the southernmost of the range, correct?
It's easternmost.
Like Maryland's just the southernmost.
The easternmost, yeah.
Pennsylvania is loaded.
Yep. So they're a species and they're endangered here in Maryland.
But they're common in other states, correct?
Because it's only in the Susquehanna where we have map turtles here in Maryland. But they certainly have threats here. So,
there's some comments in the chat. People are really excited about this study and that you're building all this data and DNA sampling and chemistry of the turtles. Someone asked, how do we go about testing our personal box turtles for their microbiomes?
You can do that. You can already get your own microbiome tested, just like you can get your own genome tested to see what your ancestry is like or that of your dog. So you could do one of those. I think they're a few $100s, and you swab the turtle and you send it off. However, it's going to be tailored to humans or maybe dogs. So I don't know what the quality would be like, by which I mean you might get a lot of question marks, kind of like what we're getting, which is a lot of lists of things that live in there, and then we don't know what to do with it. But we also don't know when you get those data from your turtle, we so far we're not going to be able to tell you if that's normal or not. We don't know that yet, but that would be a great idea for some citizen science kind of stuff. To have more data from a bunch of people testing their turtles might tell us something about that.
Well, the genetics are there a lot different too, because, like mid-Atlantic fox turtles, they're very colorful, stuff like that. You go to different parts out Midwest or something, you look like almost a different species.
Yeah.
And probably crossbreeding with ornates too sometimes and cross over, but they look very different. Fox turtles look very different from one all the way down. They look very different.
And that's something we're just starting to address in humans, because the microbiome, of course, interacts with our own genotypes, and we're all different, just like our parents told us when we were all kids, we're all special. And the same is true for turtles. The microbiome in one might be different than in another, like Jace is seeing, maybe due to genetic factors, maybe due to their diet, probably some combination of the two. Just another complication. Yeah. Steve, yeah.
I had a couple questions. So for one, do you know what role probiotics would play? I thought of it first with the study with autism in humans. Then vegetables at the zoo, if they're supplemented, are they getting Nutravac or anything like that? Because I know, supplements are given to I've in all my reptile keepings, something that comes up every now and again.
Yep. Yeah, we just add that to the list of things that are part of their diet. If you're dusting the food with something.
We described the diet in the paper, right?
Yeah.
We did a full, Yeah, so if there's a description of all the veggies and the typical, right?
What are probiotics?
And then I think it's just vitamin dusted.
Okay.
Yeah.
So, but calcium may be a limitation in the wild turtles, for example. So that might be something.
You'll never be able to duplicate a wild turtle's diet with captive breeding. That's why you can mostly tell a captive bred turtle from a wild turtle, because you cannot duplicate what they eat. They eat everything.
Yep, and
corals and everything, they eat everything.
And it probably differs a lot by season. You saw differences in the microbiome with Jace's data from spring and then summer and fall. And you've been out in the woods, that what a turtle might eat is quite different in the spring.
I'm just living by a pawpaw tree or something.
Yeah.
So you're on one side of the gunpowder, they're everywhere. The other side of it, they're not.
Yep.
Yeah, it's kind of funny if you don't mind, Steve.
Oh, please go ahead.
But the really interesting about fox turtles is there's a bunch of polymeries in it. It's just I don't really come across them that often, but right here in the fox turtle ones, if they fall in, there's some on the inside, there's some on the outside, just right there. So yeah, it's maybe it was the pawpaws. The abundance of pawpaws in the area. They're both eating it, so the inside and the outside.
I could probably add to that, and I didn't talk about it in this, but another question that I had is, we see differences at a state level, even within a site or between sites, and then I kind of want to know how deep does that rabbit hole go? That if you look at one individual site, like you were saying, if there's a lot of pawpaw trees on the east side, none on the west, how, and we know these turtles have pretty small home ranges, so turtle that lives on the westernmost side may not ever in its life venture onto that east side. So if there are differences at that such fine scale, geographic scale, still again could be reflected in the makeup of the microbiome.
Yeah, and pawpaw ferment shortly after they drop too. Kind of sets that attracting. I have seen. I know some types of turtles will eat their feces. I'm not sure. I don't know if I've seen box turtles do it, but I have seen redfoot tortoises do it. I've seen iguana do it, but I imagine that they would eat any scat they see in the wild. So if you have whatever, I've encountered some kind of droppings full of pawpaw seeds. I'm not sure what it was. This is in South Carolina this past year, and I feel like that could make a difference.
Yeah.
But I was wondering if you had insight on like how parasites might impact that, like different things like hookworms or pinworms. I know a lot of stuff like in the wild lives with it and still survives, like, because it benefits the parasite to keep the organism alive unless it's a parasite. And like, I know that a lot of times with like reptiles taken from the wild, if you treat them with the flagell and panicure and that kind of stuff, it can do more damage than good by wiping out the gut microbiome. I've gotten into green tree pythons the last few years, and that's a big thing there. But nevertheless, like, how would that impact the gut biomes in different turtles? Do you have thoughts on that?
The data is pretty limited. We mentioned some of these disease outbreaks aren't parasites or viruses, but it's kind of a similar question that I don't want to say I'd love to study because there would need to be a disease outbreak, but it would be very interesting to have a before, a during, and an after. One of these really high mortality ranavirus outbreaks or something like that. In the data set that I'm working with, I was really excited at first because we had some turtles, not a whole lot, test positive for herpes virus and adenovirus, but at least as far as we know, those are host-adapted viruses, which pretty much just means the turtle can have it, they can spread it around, but it doesn't really harm them. Just kind of lives and floats around in them. And we didn't see any difference in the microbiome between a turtle that tested positive versus negative. Would be really interesting, something like ranavirus that is often fatal. What that's doing in there, is it killing off certain bacteria or promoting the growth of other ones? So.
But not all individuals that get ranavirus also die.
Yeah,
That's what I was wondering. Like, you could do a ranavirus study or hitch a ride to a ranavirus study, since it's fatal, so it's really tough to do a study on unless an outbreak is actually happening, and see if there's any differences between the ones that do not make it immediately die off, and then there's some that just pull through for some reason, and it's really the individual difference thing.
Sometimes it's the ones that are co-infected with other things.
Right, that's the other factor in that. I forgot about that. Like other virus, like herpes virus is in one of them,
Tom.
Years ago, Sandy Barnett talked about site-specific immunities to diseases, and that she didn't think it was a good idea if you find a turtle crossing Hamilton Avenue, pick it up and take it down the shore and let it go. What are your guys' feelings about that?
First thought is if it's trying to cross the road.
I'm talking about in the city where there's no box turtles.
And then
this is probably
relocating.
Yeah.
Probably leave it where it is. It's
but you can't leave it in the inner city where there's no grass or anything. It's obviously escaped from somebody's yard.
I see.
Yeah, that one. Yeah, so with box turtles, there is not much fine scale population structure. So why that's important, like let's say we know a lot about this with African elephants. We know if a bunch of ivory shows up in Southeast Asia, you can kind of figure out which country it came from based on its DNA. With the box turtles, you can't figure out very, very well where they came from. Basically, there are two populated. You saw the map early on. They have a really big range, eastern United States, but they're basically like two populations. So if you find one here that's like a released pet, and you don't know where it came from, it probably shouldn't be released again, because it might have disease, and it's probably not going to be locally adapted to.
That's what she was saying.
The things that are out there. But if you find one in Western Maryland, if you know it came from Western Maryland, you could probably return it to suitable habitat in Eastern Maryland. That would probably be okay. For box turtles, for like bog turtles, that would probably not be the case because they have much smaller populations. So it means you got to know a lot about the turtles.
Speaking of other turtles, are there any other microbiomes like done on other turtles, or is it just box turtles?
There's a little bit done in blandings. There's one study in the lab on three-toed box turtles, but the microbiomes were much different, I think, because they were not raised on dirt. We've compared them to these turtles.
Oh, yeah.
And there's this whole phylum of bacteria, soil bacteria, that are present in these turtles that were not present in those. Probably because they were being raised on, like, something like that, sterile mulch or something like that. Haley's done the map turtle stuff. We're trying to get that published right now. That should be out in 2026. And a fair amount of work on sea turtles. But for non-sea turtles, just a handful of species, and almost all of them are in this family.
Yeah.
Blandings box.
How different is the map turtle research? How different is the findings, I guess, or even like the research methods that the box turtles?
The methods are very similar, which is good because the findings are much different. Since they're probably because they're a riverine turtle, what we find in their mouths and in their GI, their cloaca are quite a bit different. Their diets are different, the environment's different. It's hard to pick out what's driving that, but we're not seeing a huge amount of similarity.
Well, I guess, I mean, not just like the microbiome itself, because I understand, like, that's understandably different, but like as far as the like, do you only do it in that one region kind of for the map turtles, or do you have other sites you do too as well?
Do you mean region of the genome or region?
No, sorry, region,
geographic region. Right. Yeah, it would be cool to compare the box turtle microbiome up near the Susquehanna to the map turtle genome, the map turtle microbiome, because they're in the very similar areas. And we do see box turtles up there, but we haven't done that yet.
I was thinking like, or if you were to compare like the map turtle biome there to like one near Pittsburgh.
Right. Yeah.
If you would find like that there was like a big variation difference, or like do you find the same thing, like similar microbiomes in the same turtles in successive years in map turtles?
We're getting to that one, to the successive years, because we do have recaptures every year and we do have swabs from the recaptures. We're just trying to get the money to do the swab, the sequencing. We do have a few samples from Pennsylvania up above the next two dams, but it would be great to have swabs from below the Conowingo and then above the next three dams.
Yeah. They're all the way above Holtwood and everything like that, all the way up, the way up to Susquehanna.
Yeah, all the way up and at least
that's
into
closed. That couple three dams, that's closed water. They can't get. They're trapped up there, and they can't go. Centuries ago, before the dams were built,
right,
but not now. Closed systems.
Yep. The radio telemetry that Rich Siegel did on the map turtles 10 years ago or so supports that idea that they do not cross the dams. Now, in a big storm, a few might wash down, but probably not up. But yeah, I would love to do that study because some of Haley's data are showing that there are significant differences in the microbiome above and below the Conowingo Dam. So right above the dam, it's the pool. There's lots of sediment, all the heavy metals, and who knows what is in that sediment. Nobody really wants to know. Whereas the turtles below have, except in storm events, probably a slightly cleaner environment. But we are seeing significant differences in the microbiome and in the metabolome. Metabolomes are all the little molecules that the microbes make. So we're not looking at the DNA, we're looking at all those little chemicals that they make. We see significant differences there too. So stay tuned. Both of those papers should come out later this year. But the dam continues to be an interesting factor for map turtles in here in Maryland.
So someone in the chat also asked, should wildlife rehab centers be testing their box turtles for DNA and microbiomes?
Yes, and then send us the samples, and we'll put them in the freezer until we know more about what we'll.
Someone is volunteering in a rehab turtle, so.
Yeah, I mean, the swabs need to be sterile, but every COVID kit has sterile swabs. Those are exactly what we use. Those tiny little ones, those little swabs. So that's really kind of the most important part. And of course, the swab needs to be kept sterile and put in a sterile tube, and then it can kind of go in the freezer, which is what we would do if we got a hold of it. We would put it in the freezer until we know more about what's normal and what's not. And then in 5 years or so, we can pull them out and say, oh, this turtle looks more like a captive turtle, or maybe it looks more like a wild turtle. Since microbiomes change over time, if the turtle has been in captivity in a wildlife center for a while, it's probably going to look more like the other organisms in that wildlife center than it would like a natural turtle, a wild turtle, would be my guess. But hopefully we'll get to the point where that could be a useful test. And the technology is coming along, the statistics are coming along. This takes a lot of computer power because we get such big data. That's coming along. So they're welcome we'll send them a kit with swabs and tubes and gloves and instructions if they want to do that. Because there are so many box turtles and so many nature centers across the range that I think that would be an interesting study.
I thought they had a DNA test to be able to tell the locality of a lot of turtles, like that they can tell where they come from. Like there was a confiscation in New York or something, I think, and they tested them and found out they come from a certain area. It matched it. I read it a while ago, like last year, but I thought they were making progress towards finding out the locality with their DNA.
It depends on the species. With the box turtles, it's not very specific, not state specific, at least. But with something like bog turtles, where the populations have historically been more separated, that does work. I know that's available for, I think, spotteds, definitely wood turtles.
Yeah. Those were part of a certain watershed. Yeah. Not only Maryland, but a certain watershed. But I think it's because of the way wood turtles are.
It was Virginia, maybe Western Maryland, Virginia area or something they come from. I think that's what they said.
Yep, with so far that holy grail has been elusive for box turtles. But again, stay tuned. The technology continues to improve. I basically asked that question as part of my grad school and found basically two populations. Now there are some new techniques that are a little bit more precise, give a little more resolution.
Somebody said go out and document.
Right.
Take them from here, here, and document everything, and that's what's got to be done, I guess.
Yep. Yeah, I used over 1,000 box turtle samples for my work when I was in grad school, where I had the sample and GPS coordinates so that I could get at that kind of thing. Yeah. It takes up about half of a freezer. Still have most of them. Because, the technology is coming around, and so we can do more stuff with that old DNA. That's another project to put on the list of future projects. There's always more stuff to do, especially in turtles, where just not as many people working on turtles as there are humans.
Not much money though.
Yep. Oh yeah.
Is there any crossover between like the microbiomes and turtles versus other herps and amphibians, or is it?
Yeah. They're again, this depends a lot on the microbe. So some of them are like all vertebrates will have some particular types of microbes. And then there are other microbes that all reptiles seem to have. And then there's some microbes that river turtles seem to have. Some that would only be shared by maybe a box turtle and gopher tortoises, say, because they're more terrestrial. And it all boils down to kind of what they do. And these kinds of clues can help us get to that, which is what are they doing when we find out where they are. So the answer is yes and no. There's definitely some overlap in the Venn diagrams. But if anybody's looking for a project, you could do a totally computer-based graduate school project where you used all these, many of these data are online now. So anybody in the world with an internet connection can get these microbiomes and they could download those and do a bunch of statistics and answer some questions like that.
Would it be all using RStudio?
Using R and some other microbiome kind of like projects. You can, it would be pandemic proof, right? You never have to come to the lab. And a lot of the projects in my lab are actually just computer projects. All the ones here, we've showed you the field methods and the lab methods and the computer methods. But sometimes we just get, like Jace's project is, we just got sequence data from Matt Allender's lab at the University of Illinois. They did all the field work and the lab work and the sequencing. So,
What's a good turtle dog go for these days?
Think about starting our own.
No, I'm just curious. I mean,
I don't know that John Rucker ever
think about
sold.
training some up.
Yeah.
I've over the years I've learned that there are a bunch of different species that will do it. Labs sometimes do it, especially if they've been trained to be nosy. One of the best turtle dogs I knew was a DNR dog in Indiana. It was trained. It was the dog that when someone disappeared in the woods, maybe to suicide, that was the dog trained to go and find the body. And the handler said that that dog, when they were going out in the woods, it would see the turtles and note them, but would keep its nose to the job. And once it found the body and it was off duty, it would go back and find those turtles. So it just naturally kind of did that. John Rucker actually breeds them for that, though. And I don't know if he's ever sold them. He probably would way undersell what they are.
It's my understanding that in Mouse and Birmingham, where mountain tortoises are eaten by humans, that was their demise, is that they trained dogs to find them, where a human could never go into that jungle and find them.
Yeah, they are a powerful tool in wildlife science, not just for turtles, but they could also be a powerful tool in the hands of a poacher. Yeah. But yeah, more than once I've played with trying to get my own, more than once I've thought about getting my own dog and training them. But to hear John Rucker tell it, you have to kind of find a dog that's naturally interested and then train it. He's got this kennel that he's had for several, I think 4 generations. And he says not every dog is interested, even though they're supposed to be bird dogs. Some of them, he just has to give them away as pets because they don't care about finding the turtles. But others just take to it.
Trained hunting dogs.
Yeah.
Thousands, $10,000, $15,000 for a trained duck dog.
Yep.
Imagine something
That's
specialized
one of
like
the
that.
turtle dogs to go for.
There was a comment that I missed in the chat. Someone said, I've read that young box turtles eat more animal diets than other turtles that eat more vegetarian, and they're wondering if that affects the microbiomes.
Yeah, probably the diet has a strong effect, especially on the cloacal, the gut microbiome. We know that in humans. If you're vegetarian, your diet's going to be much different. If you move in with the roommate and start eating what the roommate eats, your microbiome is going to change. So it makes sense that the same would be true for pretty much anything with a gut microbiome. We haven't done that experiment yet, but again, that would be a great experiment to do with captive turtles where you can control their diet and see what happens. So it might be another good citizen science project that we can put on the list of things we need to learn about turtles. Right? Because you could go test turtles and learn about the environment. Say maybe this habitat is deficient in animal protein or something. And we know that because some bacteria is missing. Something like that. Anything else?
I have a question about pesticides.
Okay.
Are you seeing the pesticide traces all season long, or are you only seeing it certain times of year? Or is it like living permanently in turtles or something?
That atrazine one showed up in turtles that were sampled in June, which is a high season for atrazine application in agricultural areas. That was the only time that those turtles were sampled. So another great question would be, and we're working up to doing this in the map turtles, because in a river that collects all some of the same turtles, but at the same site, spring, summer, and fall, year after year. So we have the statistical power to maybe address that. So we'll see what happens when we get to that stage.
Some are right up against the cornfield.
Yeah,
Illinois.
Like within spray. What do you call that? When
Overspray.
Yeah, within overspray distance.
I read somewhere where every living thing's got the chemical. We got it in our
That's
body.
what
right now.
that's my
Everything's got it.
understanding.
Yeah.
What's it called?
Glyphosate.
Glyphosate.
Yeah, everybody's got it.
It's in most of the bread that we eat.
We're trying to get some money to study more PFASs. You may have heard them as forever chemicals that are very useful. We, as humans, have been making them since World War II, really. But they're in everything. You find them in polar bears. You find them in Arctic ice, thousands of miles from where they have been produced or used. But they move really well in the atmosphere and in water. So yeah, they're in all of us too. So when we do studies, we like to have a study group and a control group, right? Nowadays, with things like PFASs, we no longer really have a control group. I'm thinking of this study in Australia where they were studying the effect of PFASs on the microbiome, and there are no turtles that don't have PFASs. It's just a matter of the quantity now, because these PFASs are so ubiquitous. And so that's one of the things we're kind of trying to trace down with these long-term study sites where the turtles are in decline for unknown reasons. It could be some pollutant. So they're really expensive to test for, but we're going to start testing for as many pollutants as we can. Yeah. All right. Well, thanks, everybody.